Okay, hi everyone. Good morning, good afternoon, depending wherever you are. So today's talk is going to be by Professor Alberto Perez. So he is the leader of the molecular recognition group in La Florida or the University of Florida. And among his research interests, well, we all know that he's kind of the key on protein-protein, protein-DNA interactions, and as well as structure prediction and dynamics of biomolecules. What is interesting is one of his research articles. I just checked it before. It has over 1,800 citations, so it's a refinement of the Amber force field for nucleic acids, improving the description of alpha and gamma conformers. Moreover, he also has a very interesting TED Talk in YouTube, so I recommend you to check it out. It's called "You Cannot Always Blame Your Parents." And now the floor is yours. Please go ahead. Sorry, Alberto.
All right. Thank you. Thank you so much, Adolfo. And thank you, everyone, for being here on Zoom today. So I have two stories to tell you about today. So one will be about nucleic acids interacting with proteins. The other one will be about predicting RNA structure from sequence. We just recently got involved into this kind of area, and we'll tell you about what kind of things we're doing and the progress we've seen. So my lab works in the development. So it's a computational lab, and we work on both developing and applying computational tools in order to understand biology. So one of the biggest contribution, I think, from our lab is the development of integrative structural biology tools. Throughout the talk, you will see what that means. The basic idea is that how do you combine computational tools with experimental techniques that provide some kind of information to be able to predict or determine structures in a way that you could not do with either technique alone. So we work on different areas in my group. Like one of the ones that we work quite a bit is in predicting how flexible molecules such as peptides fold upon binding a certain protein. So a big part of the group works in this area. Then we work on method development, trying to understand what are the mechanisms of action that take you from one state to another, what are the routes, what are the pathways, and can we understand how this leads to, say, misfolding or any other process that we want to stop. And finally, we have another area of the group that works in nucleic acids, and this will be where I will be focusing on today. So for the talk, the way that I will lay it out, I will first give you some of the methodological details that will be going throughout the talk so that you can understand what is it that we do and how we do it. And then I'll give you a specific example applied to protein nucleic acids and then one for RNA. All right, so the basic technique that we use in the lab is molecular dynamics. So it's just the wiggling and jiggling of atoms through time. So basically you have a physics model, you have an atomistic description of your system, and what you do is you propagate the equations of motion through time. You have to integrate them numerically at very short time steps. Femtosecond, that's the time scale for vibrations. And in order to get to timescales that are biologically relevant, you have to do these billions and trillions of times, which means that you need quite a bit of computer time to do anything interesting. But at the end, what you get is like these huge ensembles that you analyze in order to understand what are the different accessible states, how these proteins behaving, and how the dynamics and structure are related to the function. So typically, if you start, say, from a native state that you know, say, from the protein data bank, and you run simulations, what you're interested in is what are the different substates that might be biologically relevant. So here I denote them with S1, S2, S3. And the idea is how are these connected? What are the timescales that get from one to the other? What are their different populations? So we worked, a while ago, we worked on this DNA clamp that can open and close. And when it opens, DNA can insert inside, and then it closes back down. And so one of the questions we had was, well, what is the mechanism of opening? Is it opening in plane, out of plane? And so that's kind of what we did in that area. But for a lot of the things that I will talk about today, we don't know the native state. We know how to model an initial system. Say, if you have a DNA sequence, you can always predict what the pDNA structure it is, or if you have a protein sequence or an RNA sequence, you know all the atoms that will be there. You don't know how they will be arranged. And so one of the questions is, how do we sample these systems using molecular dynamics techniques in order to get to the native state? And two, how do we know if we have even found this native state, or if we have not? How do we do it with a statistical significance that we can extract any conclusions about the process? And so these are the kind of things that we are interested in. Of course, many of you use molecular dynamics in either coarse grain or atomistic models. And one of the issues with molecular dynamics is that it has very frustrated energy landscapes. So that means there's a lot of minima, and every time you fall in a minima, the simulation will spend a lot of time in that minima until it's finally able to explore other regions of the conformational space. So this is a problem that we call exploration versus exploitation. So exploration, that would be your ability to broadly sample phase space, getting over these energy barriers. Whereas exploitation is once you get to a minima, the system stays there for a long time. And so when you have exploitation, that tells you that this is a relevant minima. However, the timescales that we're able to simulate, we don't get enough exploration. So we don't know if the minima that we have found is the best one, or if we are very far away from it. So a lot of what we do is in changing this balance between exploration and exploitation. There are multiple ways of doing this, so you can use different enhanced sampling tools and whatnot. One of the things that we and others realize is that using information can help you focus on relevant regions of the phase space. So if you have a lot of, say, experimental data, say from NMR or crystallography, then you don't really need much simulation. So here on this plot, you would be on the bottom right. And then what you would get is that with very little sampling and with very coarse physical models, we are able to identify the native state. On the other hand, if you don't have that much data to help you out in the sampling and of phase space, then you are at the other extreme, where no matter how much simulation time you use, you may never get to sample the native state. And so what we specialize is somewhere in between, where we have some information that's not enough on its own to predict the structure of the native state, and we have our simulations, and when those two things combine, we're able to sample or determine the structures. So a lot of the data in this intermediate region is going to be data that it's either sparse or ambiguous or noisy. So ambiguous means there are multiple interpretations of the data, only some of them are correct, and noisy data means some of the data that you get is correct and some of it is incorrect. And so ideally, when we use this data, we would like to sample an ensemble that now has information about the native state. And this can be framed very easily in terms of bias and inference. So what we like to sample is this posterior distribution, which is the ensemble that we're going to analyze. And that comes from two contributions. One is our prior, which is given a force field. That's the Boltzmann distribution based on the force field. And then we have the likelihood that the structures that we are sampling satisfy some subset of the data. We cannot satisfy all the data at once, since it's noisy and ambiguous. That would lead to errors. So the prior is shown here as like the energy landscape. The likelihood, we usually encode it as flat bottom harmonic restraints. So the idea is that if you satisfy some subset of data, then those conformations are not going to have any restraint penalty. Anywhere else that doesn't agree with any subset of the data, they're going to have a large energy bias that's going to lead you to these other regions. So in that way, we cover phase space more rapidly. We exclude the large regions that do not agree with any subset of the data, and we just focus on the sampling on the regions that are compatible. And so when you combine these two things, you get the the energy landscape that you're actually sampling from, so this posterior distribution that you're going to get. And what you see is that if I said that our initial force field has a frustrated energy landscape, this potential is even more frustrated. So you have very deep minima. It's going to be very hard to get around from one minima to another. So to get around this, we use techniques based on replica exchange. So the idea is we're going to have multiple copies of the system. We're going to change the Hamiltonian and the temperature. So at the highest replica index, we are simulating at high temperature so that we have more kinetic energy to go over energy barriers. At the same time, we have vanishing restraints. So your energy landscape is exactly the same as what your force field likes. Whereas at the lower replicas, we enforce the subsets of data very strongly as strong restraints, and then we have a low temperature. So you're going to basically sample different minima in the energy landscape. And so in between, you are kind of modulating how strong this Hamiltonian is. So just a simplification of this view, if this is my energy landscape, and I have two regions that are shown here in gray that can be compatible with some subset of the data, what I'm going to add is flat bottom harmonic restraints so that they cover this region. So when I combine those two energy contributions, what I get is a more funneled landscape. So what you are seeing is that the regions that do not satisfy either of the gray regions now has a much higher energy, and so that leads to rapid, in this case, folding or binding into structures that are compatible with the data. Now, what is really cool about this is that the energy in the gray regions has not changed. So why is that important? Well, that is important because then the ratio of populations in those gray areas, 1 and 2, will be exactly the same as they were in the original force field. So you're basically getting a different partition function now, but the Boltzmann weight in region 1 and region 2 is exactly the same as in the original simulations. All right, so that's kind of the methodology that we're going to be using. A couple of things to note about this methodology. First, we're going to be using replica exchange. Replica exchange depends on the number of particles. So most of the time, we're going to be limited to using implicit solvent. So we have developed with Carlos Zimmerling version of the generalized Born NEC2 for nucleic acids. So it allows us to run simulations with both nucleic acids and proteins. It's really fast. It's compatible with the FF14SB side force field and the PARM-BSC1 force field. So the other thing is that when we are doing the replica exchange, what we care about is how much is the overlap in energy distributions between replicas. That will allow us to have successful exchanges between neighboring replicas. When you have a phase transition, such as folding or binding, there's a gap in the energy distribution between the unfolded states and the folded states, or the unbound states and the bound states. So that means is that you have very frequent exchanges along the unbound, very frequent exchanges along the bound states, but very infrequent exchanges in between. And so this is something that we have to remedy in order to get more efficient sampling. So just as an example of what that would look like, so this is a trace from a simulation in which I'm showing you for a particular walker in the replica exchange, which I'm drawing in cyan, how it goes up and down the replica ladder. So every time it goes all the way up, it's going to the highest temperature. Every time it goes to the bottom, it's going to the lowest temperature. And so by doing these round trips, it's sampling all possible conditions, and it's being able to get advantage of this replica exchange kind of protocol. So we want as many round trips as possible. However, in many cases for binding and folding, what we see is local exchanges. So that means if you look at this gray replica walker, for example, at the top, it is never coming down to the bottom replicas. So that means there's no efficient exchange. So your replica exchange is really not helping you out very much. If you look at the local exchanges, though, you would think, oh, this is exchanging very well. because locally it is exchanging with the upper and lower replicas very efficient. So one way of quantifying this is by looking at these plots, which tell you, the red line tells you what is the probability that if I'm at the bottom replica, I will make it all the way up to the highest replica. And so as you advance in which replica you're at, you find that the probability of getting to the highest replica should increase. So whereas the black line shows you, if I start at the highest replica, what is the probability that I will ever make it to the lowest replica? So you want these lines to be as close to the diagonal as possible. And what we see is that for systems that do not obey these kind of things, we see these sigmoidal curves at the bottom that tell us there are issues. All right, so those are the things that we care about when we do the methodological developments. This is not something that I'm going to show you more throughout this talk, but it's something that it's where we are doing our method development efforts. The other place where we're trying to make developments is in what types of data do we use. So right now we have implemented a sparsely labeled NMR. So say that you label some residues, and then those residues are the only ones that give you signals. So how do you solve these kind of structures? Cryo-EM data, we're using 5-value analysis in folding, chemical shift perturbation for binding, PREs for structure determination. And then one more thing that we use is general knowledge. So how do we frame our knowledge, say, from the protein data bank about proteins or nucleic acids into a way that it's into hypotheses that can be tested in the simulations in a way that they are ambiguous, noisy, or sparse. And so this is one of the examples that I will show today. We'll use this general knowledge. All right, so that's about the methods part. I'm going to now tell you a little bit of a story about, this is work that Reza, who is here in the audience, did on predicting protein nucleic acid complexes. So the idea is that when proteins bind DNA, they can induce a large conformational change in the DNA. So some of the DNA will remain as pDNA structures, and such in the case of BZIP. Some of them will have a slight bends, like P22 introduces 2 bends in DNA, and the Tata box here introduces a large kink on the DNA. Now, when you start from the DNA sequence, you don't know how much this structure is going to deform once the protein binds. And so what we found is that a lot of the methods that are out there for, say, docking, for predicting complexes of protein-protein or peptide-protein are not really suitable for predicting protein-nucleic acid interactions. Part of it is because these systems are highly flexible. Part of it might be because the deformation of the DNA is too extreme and we don't know how to model that. So I think Haddock uses normal mode analysis in order to predict the deformation of DNA, and then they try to dock the proteins to the different conformations they get. But then it has the issue that you lose some of the sequence-dependent properties of DNA. So what we wanted to do is, can we go beyond that by using these kind of approaches based on molecular simulations and information, this MELT approach that we developed? The other thing that we wanted to know is, well, some of the proteins that bind DNA, they bind to very specific sequences, and some of them will bind very promiscuously. So they will be able to bind many DNA sequences. So part of the explanation for this is that in some cases, the protein-DNA interaction is driven by what we call base readout. So this is the protein making specific interactions with the DNA. And in other cases, it's guided by this shape readout. So the shape readout is, what is the ability of a particular DNA sequence to adopt these bound conformations that might be extremely bent? So not all sequences can adopt those conformations. So at the end of the day, what we want to do is, if I give you a protein and I give you a DNA, then I should be able to predict multiple binding and binding sites. And by analyzing my trajectory, looking at the highest population cluster, I should be able to say what the native state is. So, as I said, we're going to be using this generalized form implicit solvent. implicit solvent is not ideal for charged systems, but we are limited by the number of replicas that we can use. So typically our simulations use 30 replicas, so that means 30 GPUs. If we had to run with explicit solvent, it would be many, many more. And so with Carlos, we already showed that this implicit solvent does a decent job at simulating DNA and DNA protein complexes. So what information are we going to use to drive the binding? So the first thing we realize is that it's usually easy to detect what the binding site in the protein is. There are even some programs that can predict what the binding site in the protein is. And so you can select all the C alphas from the binding site. And then from the DNA, you can imagine that it's a bunch of repetitive patterns of binding sites along the DNA chain, right? So some of them might be through the minor groove, some of them might be through the major groove. You don't really know. So what we do is we place a dummy particle at the center of each base pair. And what we're going to do is we're going to create all the combinatorics of possible restraints between the C alphas and the dummy particles. If we have like a protein that binds in two different domains, we might say, okay, half the DNA will have to bind the upper half, the other half will bind to the lower half. And based on that information, how can we predict the protein structure? So in this process, we're not interested in observing the unfolding of the protein or of the DNA. So we're going to keep, as we go to higher and higher replicas, we're going to put some restraints that limit the unfolding of these two macromolecules. So basically, what I'm showing you here is that despite these restraints, the DNA has plenty of mobility. The protein can change its conformation quite a bit as well. But then the protein-DNA interaction, which is what we care about, is going to sample multiple states. Some of them will be misbound, some of them will be bound, some of them will be unbound. And so that's the green plot at the bottom. So at the end of our simulation, we can just look at the ensemble at the lowest temperature. We can cluster that ensemble, and we can select the highest population clusters as representative of our systems. We simulated 15 different systems, and I'm showing you the results here. So I'm giving you the superposition of our prediction with the structure in the PDB, and I'm also giving you three numbers. One in bold is the RMSD between the top one structure by population. So this is, I run my simulation, I look at the lowest temperature replica cluster, take the highest population cluster, take the centroid of that cluster, and do the RMSD to the native state. So that's that bold number. The second number tells us about, if I look at the top 5 clusters, how well am I doing? And so this is in resemblance to what you would do in CASP or Capri, where these are blind competitions where you submit your models and then the community assesses you. And so typically you are able to submit 5 to 10 models. So we look at the 5 top models to see what is the success in sampling. The last number tells us, is there anywhere in our ensemble where we have found a good structure? And so these numbers aim to tell us, here you have a tool that is able to sample native states, and then can you identify the native state from those ensembles, or can you not? So we have, for 10 of the systems, we have this green check mark saying that yes, we were able to sample the native state, and yes, we were able to identify it as a high population cluster. And for 5 of the systems, we were not able to do so. So some of these we already expected. So for example, I think 1GJ4 has flexible tails that insert into the grooves of DNA. So those are not easy to simulate. They're very sticky in our simulation, so they are not easy to simulate. 1BGB and 2BOD at the bottom right, those have a pocket that needs to open, then DNA inserts, and then it closes. So because we're using restraints to keep the protein from unfolding, this is not something that we can simulate. And in fact, we can't get that right. So there are limitations to this protocol, but it has been able to, starting from the pDNA, sample the bound conformations of multiple different protein DNA systems. So the next thing that we were interested in was, well, you can predict some of these complexes. Do you keep like your sequence-dependent properties, or is your data so driving the binding that you cannot distinguish how good it likes to bind different sequences? So for that, what I'm going to show you is an example with bZip. So I'm using here the consensus DNA sequence that binds this protein, and I'm showing you the top 8 clusters by population. So I'm superposing that for prediction based on the cluster to the X-ray extractor, and you can see that clusters C0 and C1, the two highest by population, agree very well with the native state. But you are still able to sample multiple other binding sites, right? Which is what we like about the method. So you are able to see some in other places along the sequence, and C7, for example, is binding through the minor groove, whereas this protein typically binds to the major groove. So we are able to sample different states. Now, I changed the DNA sequence, run exactly the same protocol, and here's what we observe as the highest population clusters. So now the highest population cluster is still binding to the major groove, but a different place in the sequence. We're also seeing like that the native-like ensembles for this sequence that shouldn't bind is still found, but at a much lower population. Okay, so now we see that we're able to predict structures, we have multiple binding modes, and also we can distinguish sequences by how much those clusters are populated. The last thing I'm going to show you about this example is, well, what is the process that we're seeing through the replica exchange ladder? So here, what I'm showing you from 0 to 29 is each one of our replicas. So the higher the number, the higher replica you are. So higher replicas are going to be at high temperature, and no data is going to be enforced. So that means that you're going to be sampling unbound states. And so what you are looking here is an RMSD distribution. And so what you can see is that as you go to higher replicas, you are seeing like pretty much very wide distributions of RMSDs. As you start annealing down in the replica exchange ladder, you start seeing that some states get populated. And so these are going to be, so if you look at 5 to 9, for example, each peak there are going to be binding sites at different points through the double helix. And as you go lower and lower in replica index all the way to 0, what you find is that the highest population cluster becomes the native one. Okay, so that was for bZip, a system for which the protein doesn't induce a large deformation. The second example I wanted to show you is this P22 binding DNA, which introduces two kinks, two small bends on the DNA sequence. So these are a couple of methyl groups that insert into the grooves. And so here what we see is that irrespective of what sequence we use, we're able to find very similar binding mode. So basically, this is driven by a shape complementarity. Given the data that we give it, the shape complementarity just comes into play, and most of the sequences are able to find the native state. There are differences, though, in how much separation between the two half-site binding sites there are. So it can be 1 or 2 bases difference. If we now look at these binding curves, what we see is that, yeah, at high replicas, you have an unbound state, but due to this shape complementarity, as you go down and down the replica exchange ladder, very soon you start converging on the same types of conformation. So the final system that I wanted to tell you about is the Tata box. And this is a system that was interesting because the DNA suffers a large amount of deformation, so it gets a large kink. And this is one of the cases where we had issues with our force fields. So at high temperatures, you are sampling unbound states. As you anneal down, you start finding native-like structures all the way down. But if you look at the RMSD distribution, at say replica 20 or 15, you are sampling more native-like states than at replica 0. So basically what is happening here is that as you go lower and lower, the electrostatic interaction between two positively charged residues on the data box that insert to introduce the kink on the DNA sequence bend the DNA more than it should be in the native structure. So basically, you have an image here of what is the data box binding protein and how much it actually brings these tails together in when it binds the DNA. So it should be around 25 angstroms, but instead it's going closer to 19 angstroms. So this is inducing a large DNA kink. And so this is something that we're currently looking at. So these are electrostatic interactions between protein and DNA. So of course, the force field for DNA has been developed independently of the protein DNA. So now you're looking at combination rules between protein and DNA that might be affecting how much the electrostatic interactions are felt. And so what some groups have started doing is modifying the van der Waals combination rules for protein DNA residues. All right, so I've shown you that for BSEP, we're able to just change the sequence and use change in the populations. For P22, I showed you that most of them bind the same, and the question remains, which sequence is the most likely to bind this protein? And so our typical MEL simulations cannot answer that question, so we develop a new assay that we call competitive binding assay. The idea is that rather than having a protein bind one sequence, you're having the protein and two DNA sequences, and so you're going to be able to bind one or the other. By looking at the lowest temperature replica and looking at the population of how much the protein is binding one or the other protein, you're going to be able to tell which one has the highest binding affinities. So in this case, the cloud of points is showing you that the orange is preferred over the blue DNA sequence. And what that looks like, if I look at it at the lowest temperature replica, so you have the protein that is either binding one sequence or the other sequence. So on the right, you have the consensus sequence. And just by playing this movie, you are able to say that, to see that the protein is spending more time binding the consensus sequence than the mutant sequence. Every time you see a jump, what is happening is that this is going to a higher replica, and then some other worker that was at higher temperature is going down to the lowest temperature. All right, so this competitive binding assay allows us to identify that the consensus sequence is the best. Can it also tell us something about this based and shape readout that I talked about at the beginning? So shape readout would be if we fix the protein, the DNA conformation, would it like one sequence or the other? And if we fix at the bound conformation, do we change the preferences for which of the sequences it binds? And so we can do this computationally. So basically, we did these experiments in which we had either the consensus sequence or a random sequence, and we fixed the conformation of both of them in the apoform, so in the bDNA form. And if we do that, what we find is that the protein likes to approach the random sequence. So the population, this part is telling you that about 70% of the time, the protein likes to be close to the bDNA random sequence rather than the consensus apo sequence. When you have one of the two DNAs in its holo conformation, so in its bound conformation, what you see is that the preference goes directly to it. So it's driven by shape recognition. So if you have holo for random and apo for consensus, it goes straight for the mutant sequence. If you go on the other direction, you have the preference for the consensus. And if both of them are fixed in its holo conformation, then it still prefers the consensus sequence. So that means that the restraint energy to keep the DNA conformation in the bounded state is lower. And when we allow full flexibility, both the sequence and shape readout effects come into play. And what we see is that about 70% of the time, it prefers the consensus sequence. We have done this with different mutants, and for all cases, we have seen that indeed the consensus sequence comes on top. We have also computed the consensus sequence against itself. So that's an internal check for the error. So if the consensus sequence is in both sides, what you should expect is about 50%. We're getting around 55 to 60%, which means that we have about 10% error in our predictions. All right. So with that, I conclude my story about protein DNA structure prediction. And I'm going to just very rapidly go into talking a little bit about what we're doing with RNA structure prediction. So this is something that I never would have thought that we would be getting involved. Like every time someone else asks me, do you simulate RNA? I always say like, well, that's too complicated. You know, many conformations, the magnesiums, and so many issues are related to force fields and whatnot that I've stayed away. But recently, I was introduced to this RNA puzzles competition by Eric Westhoff. So I was at a conference in China where he introduced these RNA puzzles. And the idea of RNA puzzles is they're going to give the community, so everyone that wants to participate, an RNA sequence. And they're going to tell you, okay, you have 2 or 3 weeks to send us back 5 your predictions with whatever method you want. And then after everyone has submitted their predictions, then everyone is going to be assessed by some people who have access to the experimental structure that it's being solved at the same time. And so the assessors do not know the identity of everyone that has submitted their predictions. And so it's a completely blind set test in the sense that the predictors don't know the structure and the assessors do not know the identity of the predictors. And so at the end, what you get is what is the state of the art of the field in RNA structure prediction. One of the nice things that was cool about this conference and meeting Eric Westhoff was that my students somehow found out that we were on the news in China. And so that made a lot of them come and want to join the group. So that was really cool. So for these past RNA puzzles, they gave us about 15 different targets. We participated in 3 of them. This was the first time that we participated. And for one of them, we actually did very good. So very good means that we got a 4.8 angstrom structure for 150 residue RNA structure that they categorized as difficult. It's a cloverleaf RNA. So what I'm showing you here is the best predicted model that we had versus the experimental structure. And I'll tell you a little bit about how we did this prediction. So we started at RNA puzzles thinking, well, if we have this melt approach where we give it some information and it's able to predict the structures by looking at the clusters, what is the information that we could use given an RNA sequence? So basically, secondary structure predictions are usually very accurate. So we're going to use that, and we're going to model them with certain ambiguity and noise. And then we're going to start from structural models from servers like RNA-Fold or RNA Composer. And so we're going to start with that. We're going to seed our replicas with those structures, and then we're going to run 1 microsecond long melt simulation. At the end, we're going to cluster, we identify the centers of the cluster, and we predict, and that's the structures that we submit. And so these were the results that we got back. So our lab participated as the Perez lab gators. So we were group number 285. That's what the assessors see. And so what you see is that the top 3 predictions by RMSD came from our group. The next best prediction was about 8 angstroms from the native state. And so if we compare the initial models that we used, so here I'm superposing the initial models from the RNA Composer, they're about 17 angstroms away from the native state. So we were confident that our method has really added some value to the predictions. So in fact, of the different predictions that we submitted, what we can see is that one of these double-stranded DNA regions is found in different orientations. So how it packs against the rest of the RNA is changing. And so the method was able to sample those kind of conformations and then predict the ones that were most native-like based on clustering. Some of the things that we learned, well, some of the things we're not doing very well. So when bases pair up, they should have like a geometry that it's mostly planar, but like our predictions, because we're using restraints to guide the RNA bases together, a lot of the times they are at an angle. And so the local accuracy is not very large. There are some backbones. So if you see the blue versus the yellow, you see that in some places the backbone is adopting conformations that are unusual. And then because this is implicit solvent, we don't have ions, we don't have explicit solvent. And so maybe refining these models with explicit solvent would have given even better results. And we've talked to Darren York to see if we can improve on these predictions. So yeah, plenty of things went wrong. Right now we're trying to look at what is our ability to reproduce this with more systems. So now we have one system that we made a really good prediction. Can we do this more systematically? And could we use other expertise from the field, like using noisy context from Barnacle or using RS to determine the accuracy of different ensemble structures and predict better structures? All right, so in conclusions. So today I've told you a little bit about the problems that we have with traditional MD in sampling processes like binding and folding. I've shown you this problem of exploration versus exploitation and how we use this MEL Bayesian inference approach to overcome these limitations by using information. And then I've shown you how for protein-nucleic acid complexes, what we're using is general information based on repetitive binding patterns along a DNA chain. And we're using also this kind of information to see like what is the preference for different sequences through these competitive binding assays. And then I've shown you a little bit about how we're getting our feet wet on predicting RNA structures. So 157 residues, if you ask me, I would never have thought that we could get something like that big to 5 angstroms accuracy. So with that, I just want to thank my group. So Reza has been leading a lot of the efforts on the nucleic acid front. Marcelo incorporated like some of the new methodologies we're going to publish on improving the replica exchange. And then everyone in the group has been really helpful in getting all these projects going forward. Thanks also for the funding agencies and you for your attention.
Oh, thank you. Thank you, Alberto. So fantastic talk. Now, now we have plenty of time. So please raise a hand, and I think you will be able to ask your question. Just unmute yourself. Okay, Pedro or Heather. Oh, many questions. Good, I don't need to ask. Go ahead, Pedro.
So, Pedro, you are muted.
Yes, yes. Thank you for the talk. It's very interesting. Yes. I wanted to ask you about the time, for instance, that it takes to predict one particular binding process. So realistic time. If I give you, for instance, some DNA sequence and protein sequence, how long it takes?
Right. So for what we did here, we have like we developed protocols. It took a while to develop the best protocols for the kind of general information we're using. But once you have that protocol, it's about a week using 30 GPUs to make the prediction. Now, what we are very interested in is, okay, we have shown a proof of concept. Now what we would like to go talk with people that work with NMR data, FRET data, EPR data. How can we use that data to make structure predictions?
I see. I think that you mentioned 32 GPUs because each GPU is solving one temperature, right?
Exactly.
I see. And just one thing, the last one, I saw in the Git repository that you use or use a combination of Conda and PIP for installing, yeah?
So those details, my students are much better at those than I am.
Okay.
But yeah, we do have, so we use the MPI just to communicate. So so you need to communicate sparsely between the different replicas. And so, so basically each replica is running on a GPU, and at given time intervals, you have to exchange information about temperatures or energies between different replicas in order to see if you make a change or not. And so the approach is highly parallelizable in that regard. But yeah.
I have some issues with installation. What is the best way to contact you? Is it possible to contact you or your group?
I mean, so the best way would be to open a ticket in GitHub.
I see. Okay,
so
thank you.
Justin McCallum will be
Yeah.
the best one to answer the technical questions.
Okay, thank you. Yes.
Okay, the next one is Simon Poblete. Hey, Simon, are you there? Okay, meanwhile, he figured out the connection maybe. So the next one is supposed to be, this is Agnes Noy, please.
You're muted, Agnes.
Should not be.
Well,
should not be.
Agnes, you're muted.
Okay. Seems I have to unmute them. Okay. No. What is Agnes? Okay. Sorry.
Okay. So yes, I really enjoy your talk. So I was wondering, so I found it very interesting. So you have all these non-specific binding modes. So is your process able to describe the binding process and how these kind of intermediate states?
Right. So that's a good question. So when I started, I thought like we might be able to do that. So where we have looked this the most is for protein folding pathways. And so at the end of the day, because you have these restraints are compatible with the end state, but they're not necessarily compatible with the intermediate states. So sometimes we see that the computational pathways that we follow are not exactly the same as you would follow for the folding process. So we have done this explicitly for protein G and protein L, which are two proteins that fold to the same topology through different pathways. And so we are able to see some differences there. What we have seen is that what you can do is if you get clusters at the different temperatures from your simulation, you can use those to seed simulations that are unbiased and do adaptive sampling and Markov state models in order to get the pathways. And that works much better.
Okay, thank you. Yes.
Yeah.
Okay, now Simon.
Okay.
Simon.
Now, can you hear me?
Yes.
Yes.
Okay, great. Yeah. Hi, Alberto. Very nice talk. Super nice. Super cool. I had a question regarding, of course, RNA, especially at the end with the predictions. So yeah, it's great that the method effectively works and can do better, let's say, the RNA composer and other standard tools. But what I wanted to know specifically is that these structures that you have predicted, do they have some kind of specific motifs that are hard to simulate, or they are more like canonical base pairs or some maybe easy to, or better to be captured with the conventional molecular dynamics context? Because, yeah, I wanted to know, for example, what can play against you even with the force field. There are some, for example, phosphate base interactions or something like that that could be more difficult to grasp.
So that's a fantastic question, and those are the kind of things we're trying to figure out. So this is on our list of what went wrong and what went right. In terms of like sometimes like we see these backbone conformations that are non-native like, and I think like that's part of the phosphates being very unhappy with you know, having to pack close to other phosphates and whatnot, and because the implicit solvent is not able to shield in a good enough way the interactions. I can't really give you a lot of detail because frankly, we don't know. So I thought we were going to run this, we were going to see that everything fails and then we wouldn't know anything. And now the question is, something has worked. So maybe this can be used. So what can we do to improve it? And some of the things that we are trying to do is go back to the protein data bank and try to learn about, okay, what are the geometries that you have, like from the pseudonodes to like the clover leaf, the holiday junctions, whatnot. What are typical geometries and how could we help maybe code this as ambiguous information? So if you have ideas, we would love to hear them out and see like if there are possibilities. We are not experts in RNA structure prediction, so yeah.
Okay. No, thank you. Oh, it looks really impressive. I mean, I didn't expect that implicit solving works so well, but it does a very, very good job.
Yeah,
okay,
I was surprised. Thanks.
Yeah.
Thank you, Simon. Do we have any more questions in the audience? If not, I will ask one. Alberto, so the way how you introduce or you use restraints to, let's say, to include some experimental information, right? So you mentioned a list of different chemical shift and some other methods. So when you prepare, let's say, the melt or the technique, do you take into consideration some specific categories and then you do refinement on the data, on the experimental data, or how does it, the model takes into account different source of experimental data?
Right. That's a really good question because that's part of where we spend a lot of the time. Like, so some data we just get from papers and then we just try to see, given this data that we are given and how it's processed, what can we do with it? On the other hand, in some projects, like, so for example, you mentioned the chemical shift perturbation assays. So those where we were working with Guy Montaglioni in some protein peptide systems, they had trouble determining their structures with NMR because they have low binding affinity. And so we started talking together and they said, well, we know they bind because the chemical shift perturbation shows a displacement. And then we said, well, let's see if we can use that. But then for us, it was a learning curve about, you know, the chemical shift perturbation, even their magnitude is very different from experiment to experiment. And so what are the thresholds that you need to use? How do you need to model this data? What is the accuracy in the data? Those are things that we don't typically know. And it takes a process of learning with our experimental collaborators about the technique, about what are its limitations, what is its possibilities and whatnot. And so right now we have a paper in revisions that it does parameter sampling on the Bayesian inference. So how much data we trust. So initially we have to learn a lot with our collaborators in order to find out what is the right amount of data to trust. Now we have a way of learning that during the simulation, which is going to make it easier.
Mm-hmm. Okay.
Yeah.
Okay, thank you. And there is one more question, Pedro.
Yes, just one doubt. How do you make sure that, for instance, in nature, yeah, there are not other proteins that are close to this complex that you are showing, and they cannot interfere with this binding process? Or
Yeah, that's a good question. That's something that we really cannot do. I guess like that's not what this method looks at, right? Like we say, given that these two proteins might interact, how do they interact? But I cannot tell you whether there's another protein that they might interact with. Like we would have to make another study with a different protein. And at the end, like you might think, well, if this protein and this DNA interact together, maybe this protein binds the other protein and you have now a bigger complex. But as you go to larger and larger systems like that, it becomes more and more challenging for us to work with these kind of methods. So these are some of the size limitations for us. Yeah.
thank you.
Okay, so any additional comment, question? So we are quite good in time. So all
Perfect.
right. So it seems
Thank you
it
so
was
much.
clear. It was very interesting, and from our side, so we say you thank you. So thank you a lot for being part of this series of seminars, and see you in 2 weeks, guys. Okay, thanks.
Thank you, everyone. Bye-bye.
Bye bye. Bye bye, guys.